What I'd like to do at this point is to kind of tie this into a television show, which you probably haven't seen. Actually, very well might have, but it's no longer on the air. But for most of the time I've been teaching here at Stick, it's been easy to talk about because it's a big one. You've probably heard of it, the show House. You've heard of House? Yeah. Oh, good. What we've done here is very much like what would happen in most any episode of House, up to a point. So the standard format of House is it starts off with somebody going into the hospital with something really weird happening to them. And House's team examines them and runs them through all sorts of different tests. And then they go up to House's office, suite, whatever, and he stands up to the whiteboard and writes down all the symptoms and signs that they've come across in their case, which is kind of what we did. They don't write down the systems that are involved, but what they're doing is trying to find what diagnosis is at the root of what's happening here. Now, we don't have to do that because we know. The case says right up front that this is cystic fibrosis. But if this were an episode of House and they had all these things up there, then they start calling out different possible diagnoses. And the whole drama of the show is to figure out which diagnosis is right. Now, the root of trying to figure out which diagnosis is right is actually a kind of problem-solving skill, which is at the heart of medicine, called differential diagnosis. And I would love to teach this class, and in fact, at one time I taught at a different school, and I taught A&P as a differential diagnosis course. Because it's a kind of problem-solving skill that's very useful. And it's not just for medicine. We actually do it quite a bit all the time. It's really just trying to take evidence of something going wrong and applying it to our understanding of the system and then picking the best solution for what it is. And the example I like to use for this is, let's say this morning you went out to your car, you put the key in the car, turned it, and the car didn't start. Okay. Then what you have to do is figure out why the car didn't start and try to get it to start. Okay. So if the car doesn't start, what are the possible reasons for that happening? And why would that be a problem? What did that do? Kill the battery. Okay. What else?
Spark plugs.
Okay, so the spark plugs might not be working. And what do the spark plugs do?
Oh yeah,
They spark.
Yeah.
So they spark because the battery is supplying them with electricity. And when they spark, what's the point of the spark? What does it do?
They put gas.
It ignites the gas. So I use that spark plug thing to get to the other thing. The major reasons why cars don't start is electrical problems like the batteries did, or gas problems like you don't have any gas in it. Now, the way I set up this originally, I specifically left out a little information to set that up. But when you turn the key in the ignition, what happens next actually can point to whether it's an electrical problem or a fuel problem. So if it is an electrical problem, or no, I should say it this way. If it is a fuel problem, when you turn the key, what is going to happen? Yeah, some lights on the dashboard, that kind of thing will go on. You'll hear a little clicking noise, which is the starter and that sort of thing. If you don't hear any of that, that means that there's no electricity. Okay, so if you turn the key and the car's not going to start, but if nothing happens, then you can guess that it's an electrical problem. If you turn the key, some stuff happens, but the car doesn't start, then it's likely a fuel problem. Now, it's pretty unlikely that it's both, because if it's both, then the battery had to die at the exact same time that it ran out of gas. Okay. One of the things that's kind of a driving force behind any episode of House is that idea which is there's got to be one reason for what's happening. And the reason why we have this list of symptoms, besides just enumerating all the different parts of the body you're affected, is also to get at what is that one reason. Now, it would be possible to imagine, that somebody has pneumonia and they have diabetes and they have congestive heart failure and et cetera, et cetera. And we could find a bunch of different reasons for these symptoms that span all of those different systems. But we want to find something that just addresses all of them at once, okay? So car doesn't start, it's either the battery or it's the fuel, or it's either electrical or it's the fuel. It's very unlikely to be both, okay? And in what we're doing here, we want to think the same thing. What one thing ties all of these symptoms together? Now, I don't imagine that you can answer that question. That's not the point here. That's sort of where we're going, to understand what it is that ties all of these together. But there's actually a very easy way for you to answer those questions when I ask them, and I don't always ask them. But when you have a case study to look at, the underlying cause has to be the reason for the case study. Okay? Why are we looking at this case today? What's the topic for today? Cells. So the problem here has to do with cells. Now, actually, the problem here has to do with molecules, cells, and tissues. This case is great for tying all of those together. But that's often where you can look at this. Now, cells is very general. But if you look at all these systems, and especially the symptoms that we see there, the specific cells involved, or for that matter, the specific tissues involved, all do the same kind of thing. What are all of these symptoms tied to?
Blood.
One more time.
What are all of these symptoms tied to? What do they all have in common?
They're all related to the nervous system. Fibrosis.
So yes, that's in fact true. And if this were an episode of House, that would be the point, is to figure out what the diagnosis is. But we know the diagnosis. And so what we want to get to is what's underlying cystic fibrosis? Why do all of these things happen at the same time?
They have to do with the cells.
Right. But cells is a very general topic.
Yeah, I was thinking like blood cells, but that's not quite.
Well, there's not really any blood cell related things here. If there was blood cells, it'd be, anemia.
But like, it seems like vaguely, like.
Exactly.
Well, let's not start with trying to fit cells into this, but let's look at this. What's she coughing up? Mucus. Why is she short of breath? Full of mucus, okay. The cardiovascular ones are actually kind of tough. Why is she having problems keeping her body weight up?
Body is going into the production of mucus and like putting and like trying to apparently trying to fight bacteria like a virus.
Well, we want to think about the digestive function. Okay.
You said earlier that her body wasn't taking in her nutrients from the deficiency.
Oh yeah.
Right, and even if we go back a little bit further, I gave you an example of why she wasn't getting some, or I addressed why she might not have been getting some of those nutrients. It has to do with the medication she's taking.
Oh yeah, antibodies and antifungals that are eating up.
Yeah, we're going back a little bit further. The P1, the pancreolipase. She's taking that as medication because her pancreas isn't supplying it. Okay. she's not getting nutrients because her digestive enzymes aren't being produced.
Yeah, that's why I was getting at the that bacteria, because
But I want to stop you because you're going down the wrong way. Okay. Yes, you might have read a recent article that says that that's a possibility, but what's going on in this case doesn't have anything to do with the bacteria that she might do. Why she's not making digestive enzymes, why they're not available for her digestive system to do its job, goes to the mucus thing. Okay. Enzymes, digestive juices, are secreted from glands, which is true of insulin also, actually the same gland. The glands are backed up with mucus, so they can't secrete things. Or the mucus that's in her lungs is from tissue that's secreting things. What ties all this together is secretions. Okay. We go down to the GERD. It's about the secretions in the stomach lining that's causing problems. Not just the GERD, but the ulcers also. Or the sweat chloride test is because sweat glands secrete things. Okay, so the tissues or the cells that are involved in this all are cells that have to do with secreting things. So that's kind of what ties this together. Now I have here, oops, I thought I opened that. A presentation that has some pictures that I want to use. Now, this presentation is called Biochemistry and Cell Biology. I used to use this across the 2 weeks worth of stuff, what we did last week and this week, as one presentation. Then I found that very exciting case study that we did last week with the tutor thing. So I don't actually do this so much. I just want to touch on a few things in this presentation. So I want to pull that up. And if we're talking about secretions, we're really talking about things inside the cell getting out of the cell. There's things outside the cell getting in, which isn't, that's not secretions, but absorption, like absorbing the nutrients in the digestive tract. So really tying this into the cell thing is it has a lot to do with the cell membrane, that what it does for the functions of cells. So I do want to talk a little bit about what the cell membrane is. Now, another reason why I like this case here at the beginning of AMP1 is because it does highlight the importance of the cell membrane. And we're going to see a lot of the physiology in this course, not just AMP1, but AMP1 and 2. A lot of stuff is dependent on thinking about what's happening at the cell membrane. Okay? Whether it's how the nervous system or endocrine system interacts with the rest of the body by hormones or neurotransmitters that are interacting with target cells. That kind of stuff all happens at the cell membrane. Electrical activity that causes muscle contraction has a lot to do with what's happening in the cell membrane. The way that the kidneys filter waste out of the blood has to do with how cell membranes allow things to cross over. The digestive system, which is mostly about absorbing nutrients, they're absorbed through cell membranes. I mean, even if we go to the reproductive system, we can go there too. The sperm has to get through the cell membrane of the egg cell. So the cell membrane is really crucial to understanding a lot of things throughout this entire course. And I like this case because it really gets down to some things that are happening at the cell membrane. Now, this first picture here is just a general picture out of a textbook of the structure of a cell membrane. What's the primary component of a cell membrane? What's it made up of?
Phospholipid bilayer.
Yeah, so it is a phospholipid bilayer. The phospholipids are the major component. And in this picture, we see a lot of those. They're pointing out. Okay, so this box here shows the whole thing. A phospholipid has two major components. There are the hydrophilic heads and the hydrophobic tails. What does hydrophilic mean? So it translates to loves water. What it really means as a property is that it dissolves in water. And so what does hydrophobic mean?
It's not so much dissolves in water.
But it's very much doesn't dissolve in water. So-
It's just, it's like whether it, like, has, isn't it whether it has, what's it called?
Well, so if we drill down into the molecular aspect, it's
Yeah.
about polar and nonpolar bonds.
Yeah.
But when we're talking about molecules, when we're talking about what's happening in the cell, it is all about dissolving and not dissolving in water. Hydrophilic compounds or polar compounds dissolve in water. Hydrophobic compounds or nonpolar compounds don't dissolve in water. And so when we're talking about the phospholipid bilayer, it takes on this shape naturally. If you just take a bunch of phospholipids and throw them into a water solution, they'll naturally take on this configuration. It's the least energetic configuration. It's the way that they want to be. The hydrophobic tails are all tucked into the center of the cell membrane where there isn't any water. So they're in their water-free or lipid environment like they want to be. And then the phosphate heads, the hydrophilic part, is exposed to the outside. And so on one side of the membrane, the extracellular fluid, for instance, that's water. And on the other side of the membrane, the cytoplasm, that's water. So they are both exposed to water, both sides. And the hydrophobic tails in the middle are tucked away. If we really think about all of the different molecules that we deal with in physiology in this course, the vast majority of them fall into polar or hydrophilic groups, meaning that they dissolve in water, which is good because there's water all over the place. But then we have lipids, which are different from everything else. Okay? When we're talking about polar and nonpolar bonds, when an atom binds with itself, that's nonpolar because they, by definition, have the exact same electronegativity. But in that list I gave you of nonpolar bonds, there was also the carbon-hydrogen bond. Okay? Those are obviously not the same element, but they have very, very similar electronegativities, so they form a nonpolar bond. And lipids are primarily made out of carbon-hydrogen bonds or carbon-carbon bonds. So they're nonpolar, they're hydrophobic. They're very unlike all of the other compounds, which are mostly made out of carbon to oxygen, carbon, nitrogen, nitrogen, oxygen, nitrogen, hydrogen, oxygen, hydrogen, those bonds, which are all polar. I think I hit them all. Having lipids actually gives us the ability to separate solutions in our body, in our cells, out into different compartments. So the intracellular fluid or the cytoplasm inside this membrane is one compartment, and the extracellular fluid outside is another compartment. And if we look inside cells, there are organelles that have their own phospholipid bilayer, separating it into even more components. And that allows us to do something inside a cell and separate it from what's outside a cell, or to do something inside of one cell and is completely removed from what's inside another cell. But we don't want to completely wall off these different components. We want to be able to move things in and out of these cells. And so there's more than just phospholipids in the membrane. Another important component are proteins like this one here on the far side or this one towards the middle that pass all the way through the membrane. Proteins, because they're made up of different amino acids, can have hydrophobic domains and hydrophilic domains. And for these channel proteins, or the pores that we see, the center of the pore is going to also be hydrophilic. So that sort of defines a little channel or pore through the membrane so that things can get through. They have two examples here. This one's kind of wide open. The other one here that they point out is a gated channel. There's something blocking the pore. Protein shape can change, and what this does is, under certain conditions, it changes shape so that that gate opens up and it can let things through. Okay. Now, the kind of things that go through channels are small charged particles. They can be fully charged, they can be partially charged. They just have to be very small. And this picture kind of shows that with a little bit more. First off, things can move directly through the cell membrane without any help, without any channels or anything like that. But they have to be lipid soluble. They have to be able to dissolve within the hydrophobic center of the molecule. Now, in the picture here, what they've drawn is supposed to represent a cholesterol molecule. It's not drawn to scale. And in fact, if we go back to this picture here, these little yellow things in one half of the cell membrane, those are cholesterol molecules. That's where cholesterol is in your body for the most part. So this thing is obviously way too big for what's drawn here. It should be about the size of the tails of one of these things. But it's just there to represent a lipid-soluble molecule that can pass through. And a good example of this sort of thing, this is supposed to represent cholesterol, but steroid hormones are very similar in structure to cholesterol, and they do that. They're signaling molecules. They get to their target cell, and they pass right through the cell membrane inside. Okay.
Why is it called the plasma membrane?
I am not entirely sure where plasma in plasma membrane came from, other than generally speaking, plasma usually is some sort of Greek word that kind of means stuff. Best translation. I prefer to just call it the cell membrane, just because plasma has other meanings and it can often get confusing. Yeah. Right. Yeah, so like plasma is matter. Plasma is the non-cellular component of blood. Plasma membrane, cytoplasm. There's all sorts of uses for that word. So I will pretty much always refer to it as the cell membrane, which is a little bit more modern way of referring to it. But yeah, I don't exactly know specifically why plasma is used to describe that, and we won't really worry about it too much. Now, polar or charged or hydrophilic compounds that need to cross the cell membrane have channel proteins for them if they're small enough. That is green, isn't it? This green dot here is representing some ion, maybe sodium, maybe chloride. I say that because it's attracted to the positive side of the water molecule, so it has to be a negative ion. And also green is usually the color that means chloride. Anyways, this particle can pass through this channel to get inside the cell, or vice versa. If it's inside the cell, it can pass out. Down here, they have large molecules, which are very much not drawn to scale for this picture, but the sort of hexagon-looking one here is probably supposed to represent a glucose molecule or something like that, a six-sided carbon ring like that. Glucose has 24 atoms to it, and so it would definitely be 24 times larger than this chloride ion right here if we were to draw it to scale, but they didn't do that. And then this squiggly line down here is supposed to represent a protein. And again, not drawn to scale. A protein is probably going to be made up of hundreds and hundreds of atoms, if not thousands. So it would be huge compared to these other things here. But they're just suggesting those kinds of things can't pass through. Glucose or similar molecules can pass through using different types of proteins. Instead of channels, they have dedicated transport proteins, which basically pull them or pass them through the membrane with a little bit of energy.
Now, so why is the reason why glucose cannot pass through and whereas like it seems like things that looks like
Like the chloride ion there, the green thing?
the
cholesterol?
The cholesterol. Is there a reason why liquid lipids can pass through liquid lipids something is because it's that it's soluble.
Right.
You know, it's not so much the fact that it's that one is larger than the other. It's the lipids.
Well, so I started off to say only lipid-soluble things like a cholesterol molecule or something like that can pass through the cell membrane without any help. Anything that's not lipid-soluble, anything that's water-soluble, needs help. The proteins that we're looking at here would allow small things like an ion particle through. Larger things like glucose or a protein can't move through a channel because the channels aren't big enough or something like that. But there is something called a glucose transporter, which physically moves it across the membrane. But again, because it's water soluble, it doesn't go right through the lipid part of the membrane. It goes through a dedicated protein for that. Okay. Now, moving across the membrane is really a function of diffusion. And diffusion is just a general thing that's going to happen, property of substances, under most conditions. I used to do this thing that I don't like to do anymore because of what it ends up doing for me in the long run, but one of my colleagues does this in his class, and I thought, hey, what a great idea. So I brought in some cologne, and I used to spray this cologne from one corner of the room, and it would diffuse through the room while I'm continuing on with this class. And so I'd stop a couple of times. It works a lot better with more than 4 people in the room, but I'd stop every so often. I'd ask people to raise their hand if they can smell the cologne. And what we'd see is that it would sort of move across the room. So early on, only the people close to where I sprayed it could smell it. And then later on, towards the opposite corner of the room, people would start raising their hands. I don't do that anymore because I don't like that cologne. And I don't like to have it, my hands smelling like that for the next day or two. So I don't do it anymore. Plus, it was kind of cruel and unusual punishment for students. Somebody thought I'd like that cologne because it's shaped like an Erlenmeyer flask, and I'm a scientist. And I thought, hey, this would be cool, but it's really kind of awful cologne. And I'm not even allowed to have it in the house because my wife doesn't like it. But no, she didn't say that. But I don't wear it because I don't really like it, and she definitely doesn't like it. But the point there is within this room, the odorant molecules from the cologne will spread or diffuse through the air in here. And in this picture, we have the same thing happening, but in a liquid instead of an air medium. So this thing that kind of looks like a purple sugar cube is supposed to represent a dye pellet, and it's dropped into a beaker of water. And at the bottom of the beaker, it starts to dissolve. And so this cloud of purple surrounding it is the dissolved dye molecules. And as the pellet gets smaller and smaller, the cloud gets larger and larger until finally the pellet's all dissolved and the water is completely filled with the dye molecule or with the dye pigment. It's diffused through the water medium completely. Okay, it's not leaving the water. It's not able to diffuse out of the water and into the air, something like that. And it's not able to get through the walls of the beaker because the glass is impermeable. But that's diffusion. That's the idea. A molecule like this will diffuse evenly throughout an entire medium up to whatever barriers there might be. So if I sprayed the cologne in here, it wouldn't get out into the hallway because the wall and the door and stuff like that would block it. It wouldn't get upstairs. It wouldn't get downstairs because it can't travel through the ceilings of floors. It wouldn't go outside, et cetera, et cetera. If we open the door, then eventually diffuse out the door into the hallway. Okay? If we can allow it through the barriers, then it will pass through, but the barriers will keep it contained. Now, diffusion is very important for how things behave in the liquid environments that we have. But having these hydrophobic barriers from the lipids are very important. Now, the proteins that allow things to pass through make them semi-permeable or selectively permeable membranes. And that's represented in the picture here with this dotted line. Now, kind of every intro biology and intro A&P book talking about diffusion has a picture like this. And I've never actually seen a U-shaped tube like this, but the point it's making is kind of exactly where we want to go with this. So in this U-shaped tube, on one side it's labeled A, and on the other side it's labeled B. It's been filled with water, and the membrane is permeable to water, so the water is going to fill both sides equally. And then on one side, there are things dissolved, a small concentration of something dissolved there, and on the other side, there's a larger concentration. In the picture here, they just say solute molecule, but just to make it a little bit more realistic, let's call those glucose molecules. Okay. So we have a fairly weak sugar solution on the A side and a fairly strong solution on the B side. Okay. The dotted line that represents the semi-permeable membrane, the spaces between the little dashes are too small for the solute molecules to pass through. The water molecules can pass through. And so when you have two different concentrations on either side of a selectively permeable membrane, if it was possible, then the solutes on the concentrated side would diffuse over to the less concentrated side to equal things out. But this membrane doesn't allow that to happen. So instead, since the glucose molecules on B can't go over to A to equal out the concentrations, the water on the A side moves over to the B side to change it. By leaving the less dilute solution on the A side, it lowers the volume there. And by going over to the more concentrated solution on the B side, it raises the volume. And so a less concentrated solution, when it loses water, becomes more concentrated. And just like if you're making, a sauce at home and you let it simmer for a while so that the steam comes off, and what's dissolved in the water when you're making the sauce becomes more concentrated. You're cooking it down. Same idea. On the other side, where the water moves in and the volume goes up, the concentration becomes more dilute as we add water to it. Okay? And that movement of water will even raise the volume on the opposite side of the U-shaped tube so that the water line goes up. Okay? And then this last example here, which is really kind of weird looking to me, but they've put a mass on the other side that's going to weight down the water. And so the force applied, the weight of that mass that's been put on there, pushes back against the water level so that it goes back to its original height. And so the force applied here perfectly counteracts the force of the water moving across, trying to dilute the more concentrated side. The movement of water, the diffusion of water, is what we call osmosis. It's just the special word for diffusion of water. And so the force that we're applying here to kind of force the water back the other way is exactly opposite to the force of the osmosis, what we would call osmotic pressure. Okay? And osmotic pressure is an important thing to consider in a lot of things in physiology, like filtering blood to make sweat or urine or milk and mammary glands, et cetera, et cetera. So osmotic force, osmotic pressure is going to be something that comes up a lot. And it's really often because of this, that water is trying to move from a less concentrated solution into a more concentrated solution to make them equal. Okay. And again, this thing with the last picture here with the weight mass on it is very artificial. Osmotic pressure really should do this kind of thing here. And this is a very artificial set of pictures. I've never seen a U-shaped tube like this. But this set of pictures is the exact same idea, but in a more realistic setting. Now we have a red blood cell here. And these three pictures represent red blood cells that have been placed in different containers of water. So imagine we take a red blood cell and drop it into a beaker with some sort of water solution. The first beaker that we drop it into, the solution in the beaker is physiologically equivalent. So this might be like the solution that's in an IV drip bag, a physiological saline bag. The concentration of the cytosol inside the cell, the water inside the cell, and the concentration outside are equivalent. We call that isotonic, which means same concentration. So the two solutions on either side of the membrane are equivalent. It's isotonic. The next example, we take the red blood cell and drop it into a less concentrated solution. Very extreme version of this would be if we took the red blood cell and dropped it into a beaker of distilled pure water. Okay. So outside the cell membrane, pure water is as unconcentrated as possible. It's just water. Inside the cell, it's concentrated with whatever components are in the cytoplasm. When you do this, through osmosis, water from outside the cell is going to move inside the cell, just like in the U-shaped tube, trying to dilute the concentration inside the cell. Now, if we're doing it red blood cell into pure water, the water moving into the cell is going to move into the cell until the cell bursts open. Okay? It's trying to dilute what's inside the cell until it's the same as what's outside the cell. And with that very extreme difference of red blood cell into pure water, it's never going to dilute what's inside the cell to be like pure water. So much water is going to enter into the cell that it ends up basically bursting the cell. In the third example, we're putting the red blood cell into a solution that's more concentrated than your average bodily fluid. So sort of as an extreme example, let's say we drop the red blood cell into a beaker full of seawater. So it's got so much salt in it, it's a very concentrated solution. Now the inside of the cell is less concentrated than the outside. So water leaves the cell and tries to dilute the water outside. When that happens, then the cell kind of crinkles up. Just like if we sucked all of the water out of your body, then you would just sort of shrivel up. Okay? And your skeleton, your bones would be poking out against the skin. And the micrograph underneath, that's basically what we see, all the little points sticking out through in the membrane of the cell there are the cytoskeleton proteins, okay, the skeleton within the cell there. We can actually see them, like it's shriveled up onto those. Now, I kind of skipped a couple of things I want to say. At the beginning, I was saying that the two solutions here are isotonic. The other two pictures, they say hypotonic and hypertonic. The pure water, if that's what we're dropping the red blood cell in the second example, would be hypotonic or less concentrated than what's inside the cell. And then in the third example, if this is seawater that we're dropping the red blood cell into, that solution's hypertonic or more concentrated. Now,
that's really the inside, not the outside.
no, this word is referring to the outside solution. So outside the cell is a hypotonic solution.
Wait. So water. Why is water? Hypo means less than.
Hypotonic means less concentrated. So if you have a dilute solution, there's going to be more water in it, but the word is referring to what's dissolved in it. So hypotonic is going to be more pure water. Okay.
Yeah, but it seems kind of strange because we're referring to water.
No, we're referring to the solutes. We're referring to what's dissolved in it. So hypotonic means less dissolved in it, which therefore is referring more to pure water. Hypertonic means more dissolved in it. So that's going to be like all of the salt that's in seawater is a hypertonic solution relative to the red blood cell. Okay. Now, really we can describe the solution inside versus outside. So the solution that we're putting the red blood cell into in the second example is hypotonic, meaning that inside is relatively speaking hypertonic. Okay. And then on the in the opposite example, when we drop it into a hypertonic solution, what's inside would be referred to as hypotonic, comparing the two solutions together. Because it's just a matter of comparing that to something else, it's more or less concentrated solution. thinking about hypo versus hypertonic in each of these situations, water is always going to move from the hypotonic or dilute solution into the hypertonic solution or the concentrated solution. So in the middle example, hypotonic is the outside solution, so water goes into the cell. In the third one, hypotonic is the cytosol, the inside solution, so water comes out. Okay. One of my colleagues, when she's explaining this to her classes, likes to use the mnemonic, which I always found kind of silly, but I share it because it can help people, which is hypo gotta go, okay? Which means water has to go from a hypotonic to a hypertonic solution, if that helps you remember. So this is about selectively permeable membranes, what can get through and what can't. And so we're talking about the solutes here can't cross the membrane, and so the water molecules will instead. Now this is relevant, oops, I forgot about that. This is relevant to cystic fibrosis because at the root problem here, it's about water crossing the membrane to dilute solutions. Okay. Now, why does she have cystic fibrosis?
It's genetic.
Yeah, she was born with it. She got it from her parents. What did she get from her parents that gave her this disease? Yeah, a mutated gene. Okay. She got one from mom and one from dad. If she got one from mom but didn't get one from dad, she wouldn't have the disease. And if she got one from dad, not from mom, she wouldn't have the disease. She got one from both parents. Okay. Usually with a genetic disease like this, you have to get 2 copies of a mutated gene. What do genes do? Or I could say, what is a gene?
It's a section of a cell that is encoding for a protein.
Well, not a section of a cell, but more specifically,
That's DNA.
right, a section of DNA that encodes for, what was last one?
Protein.
A protein, right. And if it's mutated, what can it no longer do? What'd you say?
The job it's supposed to.
Well, yeah, the job it's supposed to, which is to make protein. So a mutation is a place in the gene that messes up the protein that's supposed to be made. Okay. Now, the gene that's at the root of cystic fibrosis is called the cystic fibrosis transmembrane regulator, or CFTR for short. This picture shows you basically what that protein is. Okay. Now, somebody that has the disease can't make this protein. They have a mutation in the gene for this protein. The protein is found in normal or non-disease states. What it does primarily is it's a channel for chloride. And that's kind of why I was setting up the cell membrane stuff earlier. So chloride is a charged particle, chloride ion, and in the cells that are affected by cystic fibrosis, normally chloride leaves the inside of the cell to get outside through CFTR. Okay. CFTR does a number of other things, which is what all this other stuff hanging off of it's for, which I don't want to get into right now. The chloride thing is the main point I want to make here. So somebody with the disease doesn't have this protein in these cells, which means that chloride does not have a way to get out of the cell. Okay. And if chloride can't get out of the cell is now impermeable to chloride, water is going to come into the cell. Okay. The inside is going to become hypertonic for chloride, and it's going to be just very slightly hypertonic. Okay. Nothing like the red blood cell examples we were talking about there a second ago, but it'll be a little bit more concentrated inside than outside because chloride can't get out. And so water molecules will come in. Now, when I was talking about things that can pass straight through a cell membrane before, I said lipid-soluble things can. Water molecules can also. And really what I should say is anything can. Lipids can very easily. Other things can very difficultly. Excuse me. So other things can be very difficult. For ions and that sort of thing, the fact that they can't get through the cell membrane very easily means that they can't get through the cell membrane. Water molecules can't get through the cell membrane very easily either, but there's so many of them that they effectively do. Let's say a water molecule can cross through, or no, let's say a charged particle of any sort can get through the membrane once in a million times. So for every million particles, one of them gets through the cell membrane. And the numbers here I'm kind of making up, but the point I'm trying to make is that it's a very rare event. If you don't have that many of those particles, then 1 in a million isn't that often. But when you have a bunch of those, so if it's 1 in a million and you've got 100 billion water molecules around, then you're going to get a bunch of water going through the cell membrane. Okay. Some cell membranes are less permeable to water, and it doesn't happen as often, but there is a slight chance of water moving through directly. Also, there are channels for water in cell membranes. So water moves across the cell membrane pretty easily. If chloride can't get out of the cell, then water is going to come in from the outside to dilute the inside solution. It's just that the chloride doesn't make it that much more concentrated than outside. So not that much water has to come in. So the cell's not going to blow up like the picture of the red blood cell we just saw. It's just going to pull some water out of the outside space. Okay. Now, the cells that we're most concerned with here are the ones that are secreting things, like the mucus in the respiratory tract, or digestive juices in the pancreas, or the proteins that we saw in the liver, those kinds of things. They're affected. Or the lining of the stomach or the small intestines. Those are secreting and membranes. And those secretions under normal conditions should have a certain amount of water in them. The mucus that you produce in your respiratory tract is supposed to be there. It helps to trap particles that you inhale, whether it's dust or pathogens or whatever. It's there to trap those things. But it's got a certain amount of water in it. It's not too thick of a secretion because you're always sort of pushing it up out of your lungs, and it ends up going down your digestive tract and you get rid of it. When the cells that are making that mucus in the respiratory tract have this problem, water is pulled out of the mucus and the mucus ends up being more concentrated or thicker than it should be. And then it's harder for the lungs to actually push it up out of the respiratory tract and clear out the lungs. Thicker mucus accumulates in the lungs, makes it harder to breathe, creates the need for her to cough up that stuff. It also provides an environment for bacteria and fungus to live, divide, and cause an infection. So those respiratory symptoms are directly because the mucus produced in the respiratory tract becomes thicker. For the digestive tract, there's kind of two things. Well, for the digestive tract, it's kind of the same thing. Digestive mucus is a little thinner than respiratory mucus, more watery, but still it gets thicker. And it makes it harder for nutrient molecules to dissolve through and get absorbed into the body. So she has a low BMI because she can't absorb nutrients very well. Also in the digestive tract, the glands that are secreting digestive enzymes, like the pancreas, they get kind of gummed up because watery secretions there get thicker too. And it's not a mucus, it's a digestive juice, but the thickness of that gums it up, which makes it harder to secrete things like pancreolipase. But then in the pancreas, it's also going to affect the ability of the pancreas to secrete insulin. So she doesn't have type 1 diabetes. It's not that her pancreas isn't making insulin. It's that the pancreas is messed up and the secretion of insulin is interrupted. So she takes insulin as a drug to replace what's not secreted from the pancreas. But it's not exactly the same thing as somebody with type 1 diabetes, which is insulin-dependent diabetes. The liver proteins that we saw earlier are primarily blood proteins. But the liver's also got some digestive function to it also. And whether it's the blood proteins or the digestive juices that it's secreting, same thing. Water's being pulled out of the secretions, so they're getting thicker, blocks the bile duct or whatever components of the liver might be there, and it's harder for the liver to produce and secrete the components it needs to for digestive or cardiovascular function. As water is being pulled out of all of these places, it has to be replaced from somewhere. And the easiest place to replace water from in the extracellular fluids is from the water in your blood, plasma. And that's going to make the blood a little bit thicker too. And that's going to lead to the high blood pressure, and the heart's going to respond to that by trying to beat faster to move the blood, thicker blood through the cardiovascular system. Sweat glands are going to be affected similarly. Okay? And so I characterized earlier that maybe her mother kissed her on the forehead when she was a baby, and she tasted kind of salty. Okay? Now, salt, table salt, is sodium chloride. Okay? And I've been talking about chloride right here. But tasting salty is just tasting sodium. Okay? So in the sweat glands, this is going on, and chloride's not getting into the sweat glands. I mean, into the sweat that's going to be secreted from them. But also water is being pulled out of the sweat. And the sweat's getting more concentrated. It's going to have a higher concentration of sodium. There's going to be a higher concentration of sodium on the skin. It's going to make the skin taste saltier. Okay. Just happens to be the table salt of sodium chloride, and we're talking about a chloride ion here. But the salty taste of the skin is really about the sodium part. It has nothing to do with the chloride.
The sodium does not go into the cell. It just
Right. So if this is a cell in a sweat gland, outside here it's accumulating sodium and ideally chloride. Chloride's not coming out, which means that there's not sodium chloride in the solution. But also because the chloride's not coming out, water is being pulled into the cell, which makes the concentration of sodium in the sweat higher, and therefore it tastes saltier. So sort of a couple of things going on there. But really, all of the symptoms that we see here, we can relate back to what's happening here. That chloride concentration inside certain cells is increasing, pulling water out of the extracellular fluid, throwing off mucus and other secretions, and that's going to basically lead to all of the symptoms that we saw there.
and in general, people with high blood pressure, not necessarily people who have cystic, but that's it.
CF is acceptable.
Yeah, right. They have having a elevated
Blood pressure?
Elevated blood pressure can be as a result of having high sodium.
Yeah. So just in general, when people eat high salt foods, they're going to end up having a high blood pressure.
Because that reason.
Well, so what's happening there outside of cystic fibrosis, the high sodium content in their food ends up getting into their blood, and then their kidneys filter the sodium out, and the water goes with the sodium, which, wait, no, I said that wrong. They want to do that. They want to filter the sodium out, but the sodium stays there, and so water tries to dilute the sodium that's in their system, and that's going to raise their blood volume. The opposite of what's happening here. Here, what I'm describing is blood volume or blood water is going down. Hydration in the water is going down, so it's thicker and harder to pump. What we're talking about here, the sodium-related high blood pressure, is because there's too much sodium and blood volume is high because you're retaining water. So, yeah, which I didn't exactly say entirely clearly, but yes, that's the answer. The underlying chemical reasons are different, but it does have to do with sodium and water and concentrations and that kind of thing. Let's see. Yeah. So this is the last picture in the presentation, because the presentation is kind of leading up to understanding what's at the root of cystic fibrosis. I want to just take a little bit of time to go back and pick up a couple other things. I really need to move the picture to another part of this. This picture here, I skipped over it. It was right after talking about dilution. I really should move it to before the dilution part. But that picture that had the glucose and the protein and they weren't able to cross over the membrane, this gets at how big things move across the membrane. More proteins than glucose, but there's two processes, one called exocytosis and the other one called endocytosis, which is a little hard to see. It's in the red here though. But endo and exo mean into and out of, respectively here. And then cyto is referring to the cell. Endocytosis is a process of the cell membrane pinching off and pulling something from outside into the cell, which is what this is suggesting here. And exocytosis is the reverse, where something inside the cell within a little vesicle is released through the cell membrane. So big things, especially proteins, are usually moving across the membrane like this. So I skipped over that picture a little bit ago. I want to go back there because as we're talking about how things move across cell membranes, it's important to kind of pick that up. Also in this presentation, we started here with the cell membrane, kind of wrapped around here. There's also this big piece here about proteins, some of which I kind of touched on I almost said last week, back on Tuesday. And some that kind of relates to what we were talking about today. This picture just shows, here we have DNA in the nucleus. It's being transcribed into mRNA, which then leaves the nucleus and goes to ribosomes to be translated. Some ribosomes are floating free in the cytoplasm, and some are attached to the endoplasmic reticulum. So there's more to this picture from the book that I got it from, which goes on to show how proteins are secreted. But here, the idea is just the DNA, RNA, protein connection, which kind of goes into the question I asked earlier about how she got cystic fibrosis, that a gene is a stretch of DNA, and if there's a mutation in it, that bad information, genetic information, goes into the process of what the ribosomes do, and certain mutations can basically mess up making the protein altogether, which is basically what happens in cystic fibrosis. This is another picture very much like the one in the book that I showed you last time about the different layers or levels of protein structure. There's the primary level of organization, which is just the covalently bonded together amino acids, and then secondary structure, which is local shapes like helices and flat sheets. Tertiary structure, which is the overall structure of a single protein strand, and then quaternary structure, which is where multiple protein strands come together. In this picture, they have hemoglobin, which is what our book has. I like this book because it has another example of quaternary structure. This happens to represent a fibrous protein like collagen, or keratin is another example. What's nice about this is that hemoglobin has 4 parts to it. And a lot of times students will say 4 parts, quaternary means 4 levels. And so they think that it's because of the 4 parts. But here we have 3 parts that come together. Quaternary structure is just when multiple things come together and they take on the total shape of the overall protein. Some proteins don't have quaternary structure because they only have 1 part, 1 subunit, or we wouldn't call it a subunit, but they just have 1 protein chain within them. Okay? But ones that get more complex and multiple things come together, that's quaternary structure. Secondary, tertiary, and quaternary structure are mostly held together by hydrogen bonds. Okay? The primary structure is held together by covalent bonds. And so when you denature proteins, did I talk about eggs last time? Whites of eggs? Yeah. So when you denature proteins, applying heat or other ways you can do it, like changing pH, the hydrogen bonds break, okay, which is going to lose secondary, tertiary, and quaternary structure. But the primary structure is intact because heat and pH or whatever is not going to break covalent bonds, just hydrogen bonds. A little bit of an oversimplification there, but basically that's the point. Part of the oversimplification is that there are a few examples of covalent bonds within these higher levels of structure, but most of it is hydrogen bonds. So if you denature a protein, there might still be some covalent connections there, but the overall shape is going to be lost. Okay. This is a picture out of yet another book. This is out of sort of the most common cell biology book, which I've never actually taught out of, but I found the picture on the web. And this is just a bunch of different pictures of proteins to kind of give you an idea of the vast or the variation in shapes of proteins. So this red one over here is hemoglobin. Down here is insulin. This fibrous protein here is collagen, which was in that previous picture. And then there's a number of other examples. This is DNA. It's not a protein, obviously, because it's DNA. But what they're showing is a protein that attaches to DNA. Or this one here that kind of looks like a sort of super pretzel. This is called porin, which is referring to a pore, and specifically porin is for water molecules to move across membranes. So these holes here are there to allow water to move across. And then there's a number of other examples, just to show you there's some variety in protein shape. And, oh, this stupid thing keeps freezing up. And then the last thing here is just some examples of how shapes of proteins and other molecules that interact kind of go together. Both of these are examples of how enzymes' active sites interact with other molecules, and it's the shape of the molecules that's important. And there's a bunch of different places that describe this a little bit. The picture on the far side with the 4 different green things on the side and the green one in the active side of the membrane, that's actually from the Encyclopedia Britannica website. That's where I got it. So if you want to see something about protein shape or enzyme substrate reactions, that's the place to see it. I think the book actually deals with it in chapter 2. We're talking about chemical reactions. But it's a good example of how protein shape is very important, how they work. If you're interested, down there at the bottom, that web address, Fold it. It's this website. It's really kind of cool if you're into this stuff. Trying to figure out how proteins fold is a very complex task. And it would be great if a scientist could just take the amino acid sequence of a gene, which we know because we have the whole genome mapped out. So we have the whole human genome map. So we can look at a gene, and based on the sequence there, we know what amino acids are in the protein that gene codes for. So it'd be great if we could take that amino acid sequence, plug it into a computer, and the computer will say, oh, it'll fold like this, and it'll have this particular three-dimensional shape. But protein folding is such a complex process that computers really don't have the ability to do that. But humans are very good at dealing with this because there's a certain set of rules, and it seems like a computer can follow rules just fine, but the rules are a little fuzzy around the edges, I suppose. So Folded is a website that was made where protein scientists can share information about proteins, And just common citizens can go to the website and sort of play it like a game. They'd get a sequence for a protein, and they try to make it fold as well as they can. And actually through this, there have been some great discoveries made on, very accurate protein folding predictions.
Yeah, when they, I really look at I don't like about it is that it doesn't give you that much like explanation of like what's going on like on a like cellular level and like a reason why like a certain cell might have used it and like a reason why this like particular cell application being done and like so like this is the reason why we're trying to find a protein for this like task and like
I imagine they don't exactly include a lot of the information because that might sort of turn some people off if they go like, oh, I got to read stuff too. So it's more presented as a game.
yeah,
If you're interested, then
that seems like that's all part of the intrigue is like I don't know.
well, if you're interested in that sort of stuff, then you just have to research the people that have posted the information. You can find something that explains that. They just don't have it on the website there. So, and then just following from there, it takes us back to the cell membrane stuff. So we kind of gone around the whole presentation there, which means that's all I have for today. Normally we, of course, would go until 4:50, but I, of course, don't want to stay around that long. I'm sure you don't either. So we'll call it a day right there. And don't forget that we actually do have Monday class on Monday, as unusual as that seems to be. And we'll be looking at tissues there.